Good afternoon, everybody. I'm Sindhu from Christu Jayanti College. I'm from second semester MSc Biotechnology. I'm going to explain about DNA replication in eukaryotics. Basically, DNA replication is process of producing two identical copies from one original DNA molecule. It is based on the inheritance. That is, it is based on the Mendelian law of inheritance. It occurs in all the living organism. Fundamental mechanism for DNA replication is same in both eukaryotic and prokaryotic, but basically in the eukaryotics, the DNA replication is more complex than in the prokaryotic organisms. So DNA replication is highly complex and it has a nucleosomal structure which needs to be unwinded before replication in eukaryotes. Majority of DNA replication occurs during the S phase of cell cycle, that is telomeres is synthesized in the late S phase. Due to the complex nature of eukaryotic chromosome, replication is not well understood as that in the prokaryotes. Eukaryotic replication is bidirectional, same as in the process of prokaryotes. Now, as we can see here, let's understand the process of replication first. Replication occurs when GC-CDK molecules is activated by the cyclin. Activated cyclin molecules activate S-phase specific CDKs, which are present in the S-phase. Then this S-phase triggers the assembly of the proteins, as this assembly of proteins initiates the replication. The first we'll discuss about the enzymes which are involved in the replication. There are majority of these are the majority of enzymes which you can see on the screen. The first one is DNA helicase. DNA helicase is also known as unwinding enzyme because it unwinds the DNA double helix structure, and it is also known as helix destabilizing enzyme. Next we have DNA polymerase. DNA polymerase binds a new duplex DNA strand by adding nucleotides to 3' to 5' direction. Also performs proofreading and error correction. This exists many different types of DNA polymerase depending on the each organisms. We'll study about the different types of DNA polymerase in eukaryotes separately. Next, we have DNA clamp, a protein which prevents elongating DNA polymerase from dissociating from DNA parental strand. Next, you have single-stranded DNA binding protein. Single-stranded DNA binding protein, it prevents the double helix from re-annealing after the DNA helix unwinds itself. So it stabilizes the double helix and it prevents them from re-annealing after the function of DNA helicase. Next, you have topoisomerase. It relaxes the DNA and in its super coiled nature, because supercoiled nature DNA is highly coiled, so it is highly very stable. After it is unwinded, it becomes unstable, so there should be something to relax the DNA during that time. That function is performed by topoisomerase. Then we have DNA gyrase. DNA gyrase, it relieves the strand of unwinding by DNA helicase. This is a special type of topoisomerase. Next, you have DNA ligase, provides a starting point for RNA for the DNA polymerase to begin the synthesis of new DNA strand. Next, we have teloisomerase, lengthens the telomeric DNA by adding repetitive nucleotide sequences to the end of eukaryotic chromosome mainly. This allows germ cells and the stem cells to avoid the nikes on the cell division. These, as I told you earlier, that there are different types of DNA polymerase in eukaryotes. There are 5 main types of DNA polymerase in eukaryotes. First one is DNA polymerase alpha. It helps in DNA replication and DNA repair. If some of the base pairs are added in a wrong way, for example, we have 4 base pairs, adenine, guanine, cytosine, and thymine. And adenine only binds with thymine, and cytosine only binds with guanine. But what happens during DNA replication? Sometimes there is a mispairing between these base pairs, so that base pairs are repaired using DNA polymerase alpha. Next, we have DNA polymerase beta. It also helps in DNA repair. DNA polymerase gamma. There are, apart from the cellular nucleus or cellular DNA, there are some DNA which is present in mitochondria also. So this mitochondrial DNA is replicated by using DNA polymerase gamma. Next, we have DNA polymerase delta, which also helps in replication of the DNA and DNA repair. And it also has an exonuclease activity from 3' to 5' direction. Next, we have replication in during the replication in mitochondria. DNA polymerase gamma also has an exonuclease activity from 3' to 5' direction. Then next you have DNA polymerase epsilon, which also helps in replication from 3' to 5' direction. So these were about the DNA polymerase enzymes which happened in the eukaryotes. Next, we have pre-initiation. What happens when there is an initiation? What happens before the replication process starts? Before the replication process starts, there's a transition of G1 to S phase of the cell cycle. Actually, during the pre-initiation stage, replication selection occurs. Replication selection is mediated by formation of pre-replicative complexes, which is called as pre-RCS. The first step in the formation of pre-replicative complexes is recognition of replicator by eukaryotic initiator, that is ORC complex. ORC complex is origin recognition complex. Once ORC is bound to these pre-replicative complexes, it recruits two helicase. Why do we use a helicase here? To unwind the DNA. Helicase loading proteins called as CDC protein. CDC protein is cell division cell protein 6, and cell division cell protein 6, and cell division TL. Together, ORC and loading protein recruits a protein that is thought to be eukaryotic replication fork helicase. So these two proteins are together called as replicative eukaryotic fork helicase. The MCM27 complexes. This entire complex is called as MCM27 complexes. Instead of replicative complexes, there are formation of GI. During the formation of GI are only activated to initiate the replication after the cell passes from GI to S phase of the cell cycle. As I explained you the process, this is how you can see on the screen that there is a replicator, and ORI, origin replicative complex, is binding to a DNA site. After origin replicative complex is binding to a DNA site in the second step, in the third step, you can see that CDC6 and CDT1 protein. These are binding together to form the complex of MCM27. After MCM27, this is how the replication start. After the replication, there is 3 steps through which the replication is, the process of replication is initiated. The first one is initiation. In the initiation, there is an autonomous replicating sequences. In eukaryote, the DNA replication is initiated at a specific site known as ARS, that is autonomous replication sequences. These autonomous replication sequences is usually 100 to 150 base pair long. There is a multiple origins in the eukaryotes, because we know that eukaryotic organism is a very complex organism. That's why we have a different replicative origin sites on the eukaryotes. It differs in the yeast and the other organisms. The origin of replication contains 3 repeats of 13 mers and 14 repeats of 9 mers. At the origin, there is an association of sequence-specific dsDNA binding sequence. The DNA binding protein called as ARS. What is ARS? ARS is autonomous replicating sequence. ARS binding factor 1. It binds to domain 3, domain B3. Domain B3 is nothing but 3 repeats of 13 mers. So these domain B3 causes a stress, and melting occurs at B2. That is second position of that 13 mers. There is a melting of the domain that occurs there. Thereby, this 2 strands of the DNA get separated. The set of proteins, such as we read about the 5 different proteins in the eukaryote, DNA polymerase alpha, gamma, beta, delta, etc. These proteins and RFC, PCNR, and single-stranded binding proteins, all these proteins collectively called as origin recognition complex binds. Origin recognition complex, this binds to the DNA and initiates the replication. ORC is a multimeric protein. The initiation of replication in all the eukaryotes requires these multimer protein subunits, which binds to the several sequence with a replicator. The two copies of the enzyme called as helicase is now bind to the protein to the origin of site to unwind the DNA. Each helicase unwinds and separates the DNA helix into the two single-stranded DNA. As the DNA opens up, there is a Y-shaped structure that is called as replication fork. Because two helicase binds, the replication fork are formed at the origin of replication. This extend in both the direction as the replication proceed, creating a replication bubble. There are multiple origin of replication on the eukaryotic chromosome, which allows replication to occur simultaneously in hundreds and thousands of numbers, because there is a requirement, there is a demand for more production of DNA in the cell. Next process is called as elongation. After the DNA initiation starts, the nucleotides needed to be added to the region of single stranded. Now you have double stranded DNA separated into the two single stranded DNA. Now these single stranded DNA, on the single stranded DNA, you need to add some nucleotides so that the single stranded DNA becomes a double stranded DNA, which further acts as a daughter cells. Now, during elongation, an enzyme called as DNA polymerase add DNA nucleotide to three end of newly synthesized polynucleotide strand. All newly synthesized polynucleotide strand must be initiated by the specialized RNA polymerase called as primer. Primase initiates the polynucleotide synthesis by creating a short stretch of RNA polynucleotide strand complementary to the template DNA strand. This short stretch of RNA nucleotide is called as a primer. Once RNA primer has been synthesized at the template DNA, primase exits and DNA polymerase alpha extend primer region with a DNA nucleotide and binds complementary to the template DNA. The template strand specifies which of four nucleotide, that is ATGC, to be added to each position along the new chain. Only the RNA and DNA hybrid primer binds complementary to the DNA template. DNA polymerase delta or epsilon extend the DNA change. Eventually, RNA nucleotide in the primers are removed and replaced with a DNA nucleotide. Once DNA replication is finished, the daughter molecules are entirely made of continuous DNA nucleotides with no RNA portion, because RNA primers are removed. So this was the second step in the DNA replication, that is elongation. Next, we have something during the DNA replication, during the second step of the elongation, we have leading strand and lagging strand which are produced. DNA polymerase can only synthesize new strands in 5' into 3' end direction. But what about the other 3' into 5' end direction? During this only, there is something called as leading strand and lagging strand which are produced during the DNA replication. Leading strand, it is a strand which grows continuously without any gaps between them, whereas lagging strand, it is formed with some dissimilar fragments between them. These are called as Okazaki fragments. These Okazaki fragments are later joined together by DNA ligase, which is an enzyme which we studied, which is an enzyme for the DNA replication which we studied. Next process is called as DNA termination. After the elongation process, the replication needs to be terminated, or else all the DNA would unwind itself to the single-stranded DNA and there will be no further parental DNA left in the cell. That's why the replication need to be terminated. In eukaryotic chromosomes, as they have multiple origin of replication, which initiates the replication almost very simultaneously, each origin of replication forms a replication bubbles, that is duplicate DNA on either side of origin of the replication. Eventually, the leading strand of one replication bubble reaches the lagging strand of the another replication bubble. The lagging strand will reach 5 ends of the previous Okazaki fragments in the same bubble. DNA polymerase halts when it reaches the section of DNA template that has already been replicated. The group of enzymes, cellular enzymes that removes the RNA primer includes protein called as flap endonuclease 1 and RNase 1. The enzymes flap endonuclease 1 and RNase H removes RNA primer at the start of each reading strand and at the start of each Okazaki fragment's leaving gap of unreplicated template DNA. Once the primers are removed, a free-floating DNA polymerase lands at three ends of the preceding DNA fragments and exceeds the DNA over the gap. However, this creates a new nicks. Unconnected new nicks are nothing but unconnected sugar phosphate backgrounds. In the final stage of DNA replication, the enzyme ligase joins the sugar phosphate backbone at each Nike sites. After ligase has connected all the Nikes, the new strand is one long continuous DNA strand without any fragments between them, and the daughter DNA molecule is completed. The daughter DNA molecule is completed, which shows that the termination of replication is completed. DNA, single parental DNA is now divided into double parental DNA. This is what I told about clamp and RNase H enzymes, which is responsible for the termination and removal of primers during the replication termination. Thank you, everyone. These are all my references.